This is Group 8's presentation on the bioreactor experiment for Chemical Engineering Senior Lab. The goal of this experiment was to determine the Monod constant for bacterial growth of E. coli using a bioreactor and shaker flasks. We used LB media to grow E. coli for approximately 15 hours before measuring the optical density of E. coli every half hour for a 6-hour time period. During the first lab session, all glassware was thoroughly cleaned with Alkanox. The bioreactor and glassware were then autoclaved to sterilize them thoroughly. The LB media was created by mixing approximately 3 liters of distilled water with 180 grams of LB media powder, and these containers were also autoclaved. The pH and dissolved oxygen probes were cleaned, sterilized under UV light, and calibrated. In addition to cleaning and calibration, the DO probe was allowed to polarize overnight before use for the experiment. The bioreactor was then assembled and double-checked to make sure everything was in order. The afternoon before the experiment, we added 60 milliliters of LB media and 0.5 milliliters of E. coli to each of the small shaker flasks under the biosafety hood. The flasks were covered with the aluminum foil caps and placed into the shaker at 37 degrees Celsius and 200 RPM in order for inoculation to occur.
On the morning of the experiment, the bioreactor was fully assembled and turned on. The temperature was set to 37 degrees Celsius and the speed to 200 RPM. The spectrophotometer was calibrated by zeroing out the machine with Then, a 2-liter bottle of LB media was poured into the bioreactor along with 50 milliliters of inoculated bacteria. 100 milliliters of LB media and 2.5 milliliters of inoculated bacteria were added to each of the 3 shaker flasks. Both of the shaker flasks and bioreactor were run for approximately 6 hours with optical density measurements taken in the spectrophotometer from each every 30 minutes. After the bioreactor run was complete, all E. coli contaminated objects were decontaminated and the bioreactor was sterilized with bleach. All countertops were cleaned with ethanol and DI water as well. The most important part of this experiment is safety. The nature of E. coli as a biological agent makes safety especially important in this experiment. Gloves and goggles were worn at all times while setting up, taking measurements, and cleaning up the experiment to prevent contamination. After use, gloves were disposed of in the designated waste container, and goggles were wiped off with ethanol. After the experiment concluded, everything was properly cleaned and sterilized using ethanol to ensure any potential E. coli contamination was dealt with. We washed our hands with antibacterial soap every time E. coli was handled, and all contaminated glassware was rinsed with ethanol and antibacterial soap after use. The bioreactor was cleaned with bleach after the experiment concluded to ensure no further contaminants were present.
This shows the semi-log graph of biomass concentration versus time for the bioreactor. The graph was expected to be linear. However, there are deviations due to limited time in trials. This is the semi-log graph of biomass concentration versus time for the average values obtained from the 3 shaker flasks. Again, the graph was expected to be linear. This graph was created using bioreactor data from a previous experiment that used M9 media rather than LB media. The data in this graph is very linear in comparison to the data that we gathered, so we used the M9 data to complete the data analysis for this experiment in order to find the Monod constant. The following equations relate parameters in E. coli growth. These equations were used for data analysis in determining the Monod constant, Ks. The rate of biomass growth is exponential with respect to time, with each instant of growth being dependent on the previous. The equations from the previous slide were used to calculate the growth parameter at each point in time. The maximum growth parameter was found to be 0.908. Then, the substrate concentration at each point in time was calculated as shown in this chart. A graph of 1 over mu versus 1 over s was created to obtain the equation needed to calculate the Monod constant. This graph should be linear, so a best fit line was added in order to obtain the equation. The Monod constant was able to be calculated with these substrate concentrations. In the M9 media bioreactor run, the Monod constant was calculated to be 0.472 grams per liter.
This concludes our bioreactor presentation. Thank you for listening, and a special thank you to Adam, Rachel, and Dr. Pascal for their help with this experiment.